We will start the cell communication journey by showing the signaling transduction that occurs in the G protein receptor system. In this slide, the purple ligand is already attached to the G protein coupled receptor, which activates it in the process. After the activation of the receptor, an inactive G protein with a GTP attached to it will approach the G protein receptor, and so the G protein is reformed so it ejects the GTP and replaces it with GTP, which activates it as a result. The activated G protein then moves towards the targeted protein or enzyme in order to activate it. In this case, our enzyme is called the PLC or phospholipase C, which has the ultimate goal of releasing calcium ions from the ER. Once the enzyme is activated, it changes shape and so causes a change of events commonly known as phosphorylation, leading to the release of calcium ions from the ER, which will be shown in later slides. In this slide, the signaling molecule detaches from the receptor, which inactivates it as a result. Once the activated G protein detaches from the enzyme, the activated G protein's GTP will lose a phosphate in the cytosol and turns into GDP, which inactivates the G protein. If the ligand was still attached to the receptor, the process would repeat itself over again. Moving on to the tyrosine receptor system, we have two individual inactive tyrosine kinase receptors which are approached by two signaling molecules or ligands. After the retrieval of the two signaling molecules, the two receptors are then bound to each other and create a dimer. The receptors are now activated as shown in this slide, and thus attract ATP molecules for the continuation of the process. The ATP molecule releases a phosphate to be attached to a tyrosine and turns into ADP. In this case, the process is repeated 6 times to create a fully activated tyrosine kinase receptor. The fully activated receptor attracts inactive relay proteins, which become activated once they are attached to either one of the phosphate tyrosine. Depending on where and how many proteins are attached, the activated relay proteins trigger different responses from the cell. In this case, if either one of the proteins is a PLC, it will trigger the release of calcium ions as a cellular response. In this slide, we can see the final step of the signal transduction pathway, where a second messenger, which is created by the enzyme, activates calcium ion channels to allow the release of calcium ions from the ER, which acts as a response to the signal. The disorder we chose to explore is epilepsy, a brain disorder where a person experiences repeated seizures over time. Symptoms for epilepsy vary from person to person, and it depends on which area of the brain is affected and what the original cause of the epilepsy is. Common symptoms, however, include having sudden emotional changes, blackout, episodes of staring, muscle spasms, and involuntary muscle movement. Some people with epilepsy often have hallucinations and strange sensations. Treatments for epilepsy commonly include surgery or medication, depending on the type of epilepsy. For example, a patient is subjected to brain surgery when epilepsies are due to the presence of tumors, abnormal blood vessels, or bleeding in the brain. Medication used to prevent future epilepsies are called anticonvulsants. Anticonvulsants reduce the number of future epilepsies. Sometimes medication can completely cure epilepsy, but other times a patient may have to take the drug for the rest of their life. So epilepsies are directly related to the functioning of neurotransmitters in neurons, specifically glutamate and GABA. We will start by showing a normal process. Now over here we have the excitatory neurotransmitter glutamate, which is emitted by the transmitting neuron going towards the glutamate receptor. After attaching to the glutamate receptor, the receptor becomes activated and opens a gateway for positive ions to enter. This process is directly related to ligand-gated ion receptor system. Glutamate is an excitatory neurotransmitter, so when it triggers positive ions to enter a neuron, it intends for a more responsive action. GABA, on the other hand, acts the exact opposite. The same process applies to GABA with the activation of the receptor and opening of the gate, but this time negative ions enter the neuron. Now, GABA is an inhibitory neurotransmitter, which makes the neuron less responsive. In a normal cell, both glutamate and GABA work in balance. In the case of epilepsy, there are more sensitive glutamate receptors and an overly abundant amount of glutamate. There is not enough GABA to balance the excitatory neurotransmitter. So over here, we see that despite there being a large amount of positive ions in the neuron, the glutamate triggers more ions to enter the neuron. With all the neurons going through the same process, cells become overly reactive and excited and thus produce epileptic discharges. Medication often helps strengthen or elongate GABA's inhibitory role in the neurotransmitter. That is all. Thank you for watching.